Hello, everybody, and thanks for joining us. And what I would like to do is to give you an update of our studies on adaptive immune response to SARS-CoV-2 in the context of natural infection and vaccination and the variants. So the first set of studies we did was asking early last year the simple question of what kind of immune response is associated with infection with SARS-CoV-2. And we specifically looked at early convalescent people that had not been hospitalized. So the logic here is to see what is the immune response that you have in someone that has successfully fought off the virus. And we specifically were interested in looking at antibody responses. Antibody responses are important in almost all currently licensed human vaccines, and antibodies bind the virus and neutralize it, so the key to prevent an infection. But also we were focused and interested in T-cell responses. CD4 helper T-cells are also critical for antibody responses because, as the name says, you need the helper response in order to develop a good neutralizing antibody response. And there also was data in animal models that suggested that CD4 T cells can also have a cytotoxic effect, just like their counterpart CD8 T cell, the killer cells. So these cells are important because while they may not be able to prevent the infection, they are really in charge of eliminating the infected cells, because once a virus is inside that cell, it's not accessible for antibodies. So the T cells have this important job of eliminating infected cells, and thereby are very important in controlling an infection and eventually terminating it. So when we ran these experiments, what we saw is that there was a good immune response at the level of antibodies, but also for T cells against spike, which was important because everybody was already developing vaccine based on spike. So it's important to have an immune response both at the antibody level and T cell responses against the vaccine. Otherwise, one would have an imbalanced response. And this is not always the case that the same protein can be targeted by antibody and T cells. For example, in dengue, that is not the case. So good news. And also at the same time, we did see T cell responses against other protein expressed by the virus. And we'll get back to that because this also has the potential, if it's desired, to generate vaccines that may have a broader breadth of responses. And finally, also, we saw T-cell reactivity to SARS-CoV-2 fragments also in non-exposed individuals. And this, we'll get back to that in the following slides. At this point, after defining what was the type of immune response we saw in an uncomplicated convalescent, we were wondering what were the correlates of protection, what are the mechanisms of protective immunity. And to understand this in more detail, we turned to the acute phase of infection. And what we saw is that really the people that in the acute phase had the less severe disease were people that had early and strong CD4 T helper responses and also CD8 cytotoxic responses. So really the strongest correlate of having a good disease outcome was not as much antibodies, but really cellular immunity. Another point that came out of this study was that the best type of immune response in terms of being associated with a less severe outcome was a coordinated response where all three types of adaptive immunity, antibodies, cytotoxic T cells, and CD4 helper T cells were coming together simultaneously, pretty much like fighting a war using both the Navy, the Air Force, and the Army is perhaps most effective. Finally, we also saw age as a major risk factor. Well, actually, we obviously it's not that we noticed this first. That was well known that age is a risk factor for a more severe disease. But what we did see is that this correlated with a shrinking pool of naive T cells that could mount an immune response against the new virus. Over the year, evidence continued to mount in terms of pointing to a substantial role of T cells in modulating disease severity and preventing a more severe outcome. As I mentioned, our studies pointed out to a correlation with T cell responses and less severe outcome. The same was seen in a study from Bertoletti's group in Singapore, where they correlated better outcomes with lower viral infections and lower viral infection in SARS-CoV-2 with a early T cell response. Data from studies with monkeys underline the role of CD8 T cells. And also in this study from Kalimudi, again from Singapore, what was noted is really that the onset of protection from the first shot of either Moderna or Pfizer vaccine was correlating not so much with the appearance of neutralizing antibodies, which tend to appear later, but with the appearance of T cell responses. Next, we turned, as the pandemic was unfolding, we turned at the question of how durable the immune response induced by natural infection is. This obviously could not be asked in the spring of last year because there were not enough cases that had been infected to a sufficient amount of time. But in the fall, we were able to perform these studies. We looked at about 200 people, and we measured simultaneously B cells, antibodies, CD4 and CD8 T cells, and asked over time, how was the immune response unfolding? Was there a memory response that was maintained at 6 months or 8 months? And the answer was yes, we did see good immune memory at about 8 months for post-vaccination. We also saw that about 5 to 10% of individuals following natural infection at the 6 to 8 months time point had low-level immune memory responses. So that really suggests that even if someone has had natural infection, should get vaccinated. In fact, people that have had natural infection and then subsequently get vaccinated enjoy very, very strong immune response, has been named hybrid immunity. The same data suggesting that there was long-lasting immunity at the 6, 8 months, a year time point was also confirmed by several different studies from other groups. So this data really underlined the fact that SARS-CoV-2 infection does induce a strong immune response, and this immune response evolves in a memory response, which is fairly long-lived, even though wanes a little bit over time, of course, as we will see. More recently, this study came out a couple of months ago. We have been able to study how durable immune responses are, not following natural infection, but following vaccination. And in this study, we looked at a clinical trial where a quarter of the dose that is currently authorized for emergency use of a Moderna vaccine was used. So what is used for vaccination with Moderna vaccine is 100 microgram. In this particular clinical trial, a quarter dose was used. We'll get back to that, the significance of a quarter dose. But nevertheless, you see here that even with a quarter dose, there are good antibody response against spike or the RBD region, and also neutralizing antibody responses that after the first and second dose, and then the response is still maintained 7 months after the original immunization, even though there is a little bit of a decrease here, about 7 to 10-fold decrease over time. We also looked at spike CD4 helper T cells and spike CD8 killer T cells, and the same picture is seen in the sense that there is a good response after the first and second vaccination, and a large fraction of individuals remain positive for this cellular immunity at the memory level 7 months after the first immunization, and it can be seen by a variety of different assays. So what is the overall picture that we see in this study, looking at all the different components of adaptive immunity as a function of time following vaccination? At the level of immune memory, as I mentioned, the antibody response is strong and is maintained over 6-7 months with a decrease of less than 10-fold. And when you look at the CD4 T cells or CD8 T cells, the same picture is seen in the sense of a good response that is maintained over time. And in fact, the decrease is about 2-fold or so for both CD4 and CD8. The question also is, how does this level of immunity compare with what is seen of natural immunity following infection rather than vaccination? And it's very comparable, both at the level of antibodies, CD4 and CD8 T cells. And I want to emphasize that this is the quarter dose. And in fact, if you compare with the quarter dose with a 100 microgram dose that is currently utilized as a vaccination regimen, the 100 microgram dose induces stronger antibodies than CD4 T cells, but equivalent CD8 T cells. But as I was saying, the 25 microgram dose is equivalent to natural immunity, which raises the possibility that sparing doses can be utilized and still induce a appreciable immune response. In fact, as you probably know, now the Moderna vaccine has been also authorized for a third dose, but at the 50 microgram level. So it is already being used in a scenario where a third administration is administered at a reduced dose. Finally, in this study, we had different groups of subgroups of individuals of different ages, so younger and older and more older people in the over 70 and so forth. And we did see that older individuals had a reduced antibody response, but had a equivalent CD4 and CD8 response. Again, very good news in that respect. Basically, I'm not going to show you any data today, but we and several other labs are running studies where we are comparing different vaccine platforms, Moderna, Pfizer, J&J, and Novavax that has performed some trials in the San Diego area. And the point here is to really not to make a scorecard, but to understand the different vaccine platform, what are their characteristics, how well do they induce the different type of adaptive immune response, and how durable these responses are. This is really, in a way, unfortunate that we have had the pandemic, but is a unprecedented opportunity to really understand how vaccine works, because never before in the history of mankind, vaccines for the same antigen have been studied with different delivery systems and different vaccine platforms in humans and have shown efficacy in humans. So really it's going to be a tremendous learning opportunity. Now, I want to go back to the point that I mentioned, that early on in the spring of last year, we had noted that there was some reactivity detected also in non-exposed individuals. And this actually was the control. So we had the people that were infected and say, well, let's, as a control, run some samples from blood donations that we had banked from 2015 to 2017, so that they could not possibly have had SARS-CoV-2 infection. And nevertheless, we saw that there was reactivity. So at that point, we and others saw that this was seen by us, was seen by groups in Germany, in Singapore, in Italy, in UK. So broadly observed. And we and others hypothesized that the fact that some people that had not been exposed to SARS-CoV-2 nevertheless had some memory T cells that recognize SARS-CoV-2 sequences could be due to the fact that these people were previously infected with common cold coronaviruses. And so they happen to have memory T cells for some sequence of the common cold coronaviruses that also happen to be conserved to a degree in SARS-CoV-2. And in fact, in a subsequent study that came out last summer, we were able to show that this is indeed the case. So in this, you have two particular T cell fragment T cell epitopes that were studied in this and shown in this graph. You see that the response is detected against SARS-CoV-2 sequence in black, but when we synthesize the corresponding match homolog peptide from common cold coronaviruses, we saw that this was recognized even better. Again, suggesting and compatible with the notion that what we're really seeing is a cross reaction, and what the antigen, the virus that induced these memory cells was indeed originally a common cold coronavirus. Now, at that point, though, we could demonstrate that there was this cross-reactivity, but we did not know whether this was good, bad, or indifferent. We hypothesized that this may be good, and that people that have this pre-existing immune memory that can cross-react on SARS-CoV-2 are an advantage because their immune system has a head start. And the study that I showed you earlier, where we looked at the development of immune responses to vaccination over time, offered a unique opportunity to answer that question. So what we did here was to basically separate the people that were in the trial in two groups: the ones that had detectable pre-existing immune memory against SARS-CoV-2, and the ones that didn't, here indicated in blue or in white. And when you look at the development of a T cell response over time, you see that the people that have this pre-existing immunity develop a stronger and faster T cell response at the level of CD4 helper T cells. So the question is now, does that translate in a higher or better antibody response? So does this increased helper function, does it help? And as shown here, it does. In general, you have, regardless whether you look at the spike or RBD or neutralizing titers, you see a faster or more vigorous on the long run immune response at the level of antibody responses. So this pre-existing reactivity, this cross-reactivity does help in the context of vaccination. And this now also has been, this data is consistent with other reports that have appeared in the literature
studies from Thiel's group in Germany that showed that cross-reactive CD4 T cells are beneficial in infection and in vaccination. A study from our group also studied healthcare workers that seem to have high level of cross-reactive T cells, and other data from Singapore and the UK, consistent with the same notion, and this study in the Journal of Clinical Investigation showing that people that had a recent endemic coronavirus infection really had a much a better clinical outcome. So we are also further interested in this notion, in the sense that this suggests that cross-reactive T cell responses can be beneficial, can modulate disease severity, and play a role in preventing severe disease. So we are mapping the immune reactivity that people have against common cold coronavirus, and then assessing which one of these reactivity, which fragments with antigen are widely cross-reactive. And this could be utilized to develop perhaps second-generation vaccines that would be broadly reactive against different coronaviruses, and for example, guarding against the potential that some other coronavirus from bats or other zoonotic source could jump into humans in the future. This could be a first line of defense. Now, we also, going back to the question of what does the immune system recognize in SARS-CoV-2, we mapped all the different pieces recognizes in convalescent people following SARS-CoV-2 infection. And you see here each line here, each row is a different component of a virus, and each column is a different individuals. This is a study of about 100 different individuals. And the take-home message here is that, first of all, each person recognizes multiple different antigens, not just spikes, so 3 to 4 different antigens on average. Then we also map within each antigen how many fragments of that antigens each individual recognizes. And very conservatively, we can estimate that one individual can recognize 15 to 20 different epitopes at the level of CD4 or CD8 responses. And so each individual has really tens of different epitopes that are recognized. And because the epitopes are presented in the context of tissue type HLA molecules that are very different from one individual to the next, is not only true that each individual recognizes tens of different epitopes, but that one individual will recognize a different set of epitopes than the next individual. This is important because it suggests that it would be exceedingly difficult for the SARS-CoV-2 virus to evolve and escape immune recognition at the population level by mutating all the different epitopes that are recognized by the population. In fact, we went on to show this experimentally, studying either people that were immunized with the SARS-CoV-2 vaccines or infected with the ancestral virus. And we studied the CD4 and CD8 response against spike in the case of the people that were vaccinated, or the whole genome in case of people who were infected. And we saw that the immune response against the ancestral or immune response generated by infection or vaccination with the ancestral sequence was equally well recognized in either the ancestral or 4 different variants of concerns that were of concern at that point in time. This was also matched by bioinformatic analysis, where we looked at all the epitopes that have been described, and we saw that the vast majority of the epitopes that have been described were totally 100% conserved in these different variants. At the same time, though, we noted this is with the fold chain reactivity against 4 different variants. And you see that more or less at the population level, there is no change. The fold change is none. It's 1, with the ratio is the same. But there are occasional donors that have a decrease in reactivity. And so we're intrigued and we're studying this in more detail, asking where these occasional donors that have a 2- or 3-fold decrease in reactivity may be associated with a more narrow immune response. So we are studying the variants in a variety of different approaches. We're part of a group of investigators that monitors different panels of different variants. We are an expanded variant panel, both at the level of bioinformatic analysis and also a network of collaborating labs where all over the world we provide our reagents, and they have been monitoring T-cell responses in the different geographical location from South America to South Africa to Europe and so forth. And also we're doing this kind of in-depth analysis that I was mentioning earlier. So far, T-cell reactivity is largely preserved, and this matches with the data we're going to discuss in a second, but the vaccine efficacy is largely retained against severe disease and death, even with the newer variants. So going back to the question of what are the mechanisms of protective immunity against COVID, clearly antibodies are very, very important. And this is the best, simplest option to have a strong neutralizing antibody response. SARS-CoV-2 is clearly susceptible to the neutralizing antibody responses, and really the antibodies can bind and neutralize the virus or prevent infection altogether. At the same time, as we've been discussing earlier, T cells also play an important role establishing a second line of defense. So they can limit disease severity and terminate the infection and really play a role in preventing severe disease and death. So the way we see this is that if you're talking about protection against infection, antibodies are really the major contributor, and T cells may play a role, but it's a minor role. But if you're talking about protection against hospitalization, severe disease, and death, then all different components, memory B cells and memory CD4 and CD8 T cells play a very, very important role. So what we really seeing is that essentially there are 2 diseases almost. One is the infection and the virus reproducing in the nasal or oral upper respiratory tract. And this is can be either asymptomatic or up to a cold or flu-like symptoms. What really is associated more prevalently, clearly with hospitalization, ICU, or fatal outcomes is when the virus spreads to the lung. This first part is fast, takes days, and that's why it's also difficult if there is not enough of an antibody response to prevent the infection. But the lung infection takes weeks, and this really provides an opportunity, if the immune system has a head start, to really prevent the lung infection. So essentially it's a race, and what the vaccine will do is give you a head start in this race and give the immune system enough time to expand the memory response and eliminate the virus before the virus can get to the lungs. This is not our data, but makes this point nicely. You see here the waning of immunity against detectable infection as a function of the time after vaccination. And still, there is good protection, but the immunity wanes, and Delta makes it worse. But when you look at protection against hospitalization and death, the efficacy of a vaccine is retained and is retained regardless of age. Booster. So this is a very topical, very current subject, and the question is, do I need a booster? And the question is that maybe this is not the right verb to say, do I need a booster? Well, in some cases, you do need a booster. And this is clearly the people that are immunocompromised, that are vulnerable for one reason or another. And no question, these people do need a third dose. But the rest of us, the question is, do we need a booster? Probably not. Would a booster be helpful? Yes. And so why would a booster be helpful in the, especially in the categories of people in 60, 65 years and older, or people that have occupational exposure and so forth? But clearly, as I showed you, the immunity is waning over time and will continue to wane over time. Even though it's maintaining its efficacy against hospitalization and severe disease, to have a booster and increase immunity would also presumably decrease the number of infections and decrease also viral spread in the population. Furthermore, there is another consideration that having a booster or further administration would be beneficial for in the context of variants. And why is that? It's basically an arithmetic calculation. So if you have that a particular variant is 5 times as hard to neutralize, but by giving a third administration, you raise the immunity level 5-fold, the two effects would neutralize with each other. Then there is another scenario which we are not seeing, luckily, with this virus, is when the virus is too different, and then you need to have a new vaccination, essentially with a new vaccine, which is what we do every year for the flu. But we're not there yet. So the booster consideration that applies to SARS-CoV-2 are really these 3 shown here. What we're seeing here is essentially that recent vaccinations protect very effectively against infection and very reducing the number of infections, and as compared to a vaccination that has been more longer time ago, where the protection from infection is decreased, but still there is a large and maintained protection against severe disease and death, which is instead what we continue to see in the unvaccinated. And with that, I'd like to conclude by mentioning that everything I showed you is a strong collaboration between the group of Shane Crotty and myself, Alba Grifoni, an instructor at the La Jolla Institute, and Professor Weisskopf, also an assistant professor at the La Jolla Institute, and many other talented postdoc, graduate student, and the Peters Lab, many cores, the clinical study core, the bioinformatic core, the cytometry core, without which we could not have done any of these studies, and many other external collaborators. And with that, I would like to thank you very much for your attention.